to talk about keystone species in your MicroHerd. And I do hope, how many people in here have cattle on their land? Oh, nice show, nice show. Everyone who didn't raise their hand, think about it. All the microbes that come out of cow's gut. I mean, you're in prairie land, right? This was all once a wild prairie, and there were buffalo there, and they created these deep, dark, rich soils together. So grazing animals are a really important aspect of soil health. But I'm not here to talk about grazing animals today, but I just wanted to mention that so that if you hadn't considered it, maybe you'll give it some thought. I'm going to talk about arbuscular mycorrhizal fungi. There are just hundreds of millions of species of organisms in soil, and you just can't manage for millions of different things. So how do you make decisions, and how do those decisions affect your soil quality and the microbes that help create and maintain soil fertility? And I'm focusing on the mycorrhizal fungi because they're the keystone species. If you manage for them, everything else that's beneficial also rises to the fore. So it improves conditions for lots of beneficials. The same choices. So you'll need a little terminology to begin with. So there's a reason they're called arbuscular mycorrhizal fungi. One is arbuscular comes from the word arbor. Arbor Day, what do you do on Arbor Day? Plant a tree. Look at that. Does that not look like a tiny tree? So that's how they got their name. And the scule, like minuscule, just means tiny. So arbuscular literally means tiny tree. These are endo. Endo means interior, inside, and the arbuscules are inside, not just inside the roots, but inside the cells of the root. And I took this picture. This is from a corn root, and this is the cell wall right here. You can see it right there. And there's 2 arbuscules inside that one cell. And then when the root is full of arbuscules, we call it a colonized root. The spore germinates. The plant actually sends a signal to tell spores nearby to germinate. They release hormones. Plants want to be colonized. Most plants form this association. Pretty much all the grasses, and most of our grain crops are in the grass family. So the plant sends out a signal upon germination, and then the spores germinate, and they penetrate and colonize the root. The plant actually guides the hyphae and creates a channel for them between the cells, and then it prepares the cells where it wants the arbuscules to be formed. The fungi form the arbuscules, and the plant surrounds the arbuscule with a membrane. Now, it's too small and fine to be seen in a light microscope like this. You know, this is all stained. Normally this stuff would be transparent, these hyphae. Hyphae are these filaments. So all these structures in actuality are transparent. But this is a very, very old symbiosis that dates back over 400 million years. Scientists believe that plants colonized land originally with the help of fungi, and that this is where this relationship evolved from. These fungi do not produce any mushrooms. Those are called ectos. And if you're ever thinking about looking at products and you see ectomycorrhiza, those are for pine trees. And unless you're growing Christmas trees, they're not going to really do you much good. They're not going to hurt anything, but you know, considering what some of these products cost, you should think twice. Make sure you're getting endomycorrhiza. 90% of all plant families associate with arbuscular mycorrhizal fungi. That's how common this relationship is. So in your mind, think everything but the brassicas. And unless you're growing quinoa, that's also a non-mycorrhizal plant, the amaranth. And the amaranth family is where we get our pigweeds, and a lot of our weeds are non-mycorrhizal. So mycorrhizal plants do well against non-mycorrhizal plants because they get an edge in the competition, whereas the reverse is true in areas where there aren't any mycorrhiza. So as our mycorrhizal populations in our soil decline, our weed problems are going to increase because we've now lost that edge that they were getting from this association. Okay, the next slide. So I'm going to give you 16 reasons you should care about AMF. This is actually a short list. There are more, but these are the main ones. They increase soil fertility. They build soil structure. They're the organisms that form soil aggregates, and that's what lets your soil have pores and be aerated. They increase the water holding capacity of soil. They produce a more nutritious product for humans and livestock. So if you take the same plant and you have one with AMF and one without, and then you do a leaf test, the nutrient content will be lower in the ones without the AMF. They replace harmful chemicals. You don't need as much pest control if you have these guys working for you. They eliminate nutrient runoff and leaching by building the soil quality, and they actually secrete a protein called a glycoprotein that glues your aggregates together and makes them water stable so they don't just fall apart and everything compresses and compacts when it rains, right? So they have to be water stable. So these guys are the main players in producing the glue that holds those particles together. They increase plant nutrient use efficiency, particularly for phosphorus. That's their most studied aspect, is they are experts at uptaking phosphorus and providing it directly to the plant. Without competition, in many ways, other than amongst themselves. Because think about it, the arbuscule is the organ of transfer between the plant and the fungus. And it's on the inside of the cells of the roots. So when they transfer nutrients to the plant, it's going through that arbuscule and then through that membrane and into the plant directly. So nothing else gets a shot at it once it's taken up. They increase essential oil production if you're growing mint, or in a lot of states now, cannabis, and medical cannabis is a big deal, so that's important there too. Protection from nematodes. How does a fungus protect plants from nematodes? Really, that one just amazed me. I'm going to talk about it a little more later. Protection from fungal and bacterial plant diseases, drought tolerance, salinity resistance, earlier flowering, more flowers, more fruit, more biomass, that's increased yields. Plus they sequester carbon in the soil. And why do we want carbon in our soil?
It drives everything.
It drives everything. Everything in the soil needs carbon. We need carbon. Every living thing uses carbon as an energy source. And it's doing harm in the atmosphere. Global warming, not a myth, guys. It's real. It's more like global change. And they call it global warming because it's actually the ocean temperatures that are rising, and then that affects the atmosphere. It's all this big interactive domino effect. But it's real. And so the only ecosystem on the planet big enough to affect that change that we're seeing now with pests that are now changing their ranges where they occur because the conditions are getting warmer, and if they need warmer conditions, they're migrating into those areas. So we're seeing a change in where plant diseases occur and what plant diseases occur in what regions as a result of global temperatures changing. And if we want to get all of the carbon out of the atmosphere where it's doing damage and into the soil, it's the agricultural system that can do it, because plants are driving that, and you guys are deciding what to plant. So actually, it's all in your hands. Pollinators prefer mycorrhizal plants. Great experiment. This guy put out all these plants. Some he gave mycorrhiza to, some not, same species. And he put them in little pots and mixed them up, and then all his students had to sit around and count every time a pollinator landed on each plant. And the pollinators preferentially sought out the mycorrhizal plants. Now, I haven't seen any research yet that explained why or how they did that. Thank you. But I suspect because insects are very sensitive to chemicals, taste, smells, that they could probably detect that the pollen might have a higher nutrient content, the sugars might be richer. That's my guess. Yes, sirree, it'll grow hair, fix your teeth, and make you taller. These guys do it all. They just do everything, don't they? So the big question here is, are they really doing all that? Are they really doing all that? And there's research supporting all that, but it seems like a lot. So how is that happening? Go ahead and, you might, oh, there it goes, it's just slow. So what you got to keep in mind is that it's actually the community of AMF that convey all those multiple benefits. It's not just one thing. They're not all the same. They're all very different. These, if you've never seen them, are mycorrhizal spores. Look at the diversity just in the spores. And our definition of what a species is really doesn't take great account of everything. So there may be far more species than we think out there doing this work. Because we look at one tiny gene, and a genome is a massive thing with, you know, millions of DNA pieces to it. So when you take a little piece of that and say, well, if this is the same as that, I'm going to call them the same species. Are they really? Sometimes they are, sometimes they probably aren't. And I think we've really underestimated the number of species out here, because to date, we have only identified 200 species. But I can take things that are supposed to be the same species and see very different behavior in them when I plant them with different plants. So I'm pretty sure we've missed the boat on that one. Yeah, go ahead, Dan. You're my button. So, to get microbial diversity in your soil, you've got to have plant diversity. And I'm going to go into details on that later this afternoon when I discuss diversity and how it works and what it does. AMF affect every aspect of plant physiology that's ever been measured. They become an integrated part of that plant. So think about that a minute. They're a part of the plant. There's nothing that has ever been tested in a plant that was not affected by this association when it was there. So that's a long evolutionary history between these guys. In fact, they create substances, chemicals, that the plant creates complementary substances, and with those substances combined, they can make phosphorus available that is not available to the fungi alone or the plant alone. So they actually have an interactive kind of integrated genome to do that. That's how much and how important they are to plants. So not having these guys in your system is kind of like, you know, hacking off an arm. How about a lung? You got two of them, you don't need them both, do you? You can survive with only one, but wouldn't you rather have both of them? You know? They're an essential component of sustainable agriculture. If you want to reduce your inputs, reduce your chemical use, reduce your fertilizers, and still get a really good yield, have your plants survive even rough conditions because these guys are protecting them. Have your plants experience less disease because they're healthier, and because these guys are protecting them, they're antagonistic towards a lot of fungal diseases. They don't play well with other fungi, which is a good thing because 80% of all plant pathogens are fungi. 80%. You don't see that many bacterial diseases. And a lot of things that look like disease are deficiencies, nutrient deficiencies. So it's hard to tell them apart sometimes. But you want to keep those microbes under control. You want the good microbes working for you, and you don't want to promote the ones that are going to cause you problems. So when you manage for your arbuscular mycorrhizal fungi, you're managing for the guys that are going to hold the diseases at bay. And the hyphae, the filaments that go out into the soil, the other microbes colonize the hyphae, just like they colonize the roots. And the plants send sugar to the mycorrhiza in exchange for the nutrients that they provide, and they secrete some of those sugars through their hyphae, and they feed the beneficial stuff. And there's chemical activity and chemical warfare going on down there in the soil when something wants to come and attack the plant. These AMF are obligate symbiotes. That means without a host plant, they die. If their plant dies, they die. They can't live without that symbiosis, without that plant. They can't get their own food. All their food comes directly from the plant and is fed directly to them within the cells. Now think about this. You've got all these plants. They're paying the mycorrhizal fungi to mine minerals because the mycorrhizal fungi can do that efficiently. And the mycorrhizae are accepting their food from the plant because the plant's photosynthesis is more efficient. So it's kind of like hiring an expert. You can do it yourself and spend 3 days at it, or hire an expert and have them fix it in two hours, right? So they're experts in their own region. They've got this partnership going. And the mycorrhiza have some control over what can reach the plant because the hyphae span all the air gaps. If you're a bacteria and you want to get to that plant and that's where all the action is, that's where all the food is, it's a long way along trying to travel through the soil. But if you can mount that hyphae and travel along the hyphae, it'll take you directly to the roots every time. So chemical warfare is going on where they're suppressing things that they don't want that might attack their host and enabling the things that are going to be beneficial, that are going to process nutrients and make them available. And as all these microbes live and die on and near the hyphae and the plant roots, they're releasing nutrients. And the hyphae and the roots both are taking them up as quickly as they can before anyone else gets them. That's the thing. So here we have all the minerals that a plant needs to thrive, pretty much. It's the basics. And we've got our primaries and our secondaries and our tertiaries. We need the most of these and a lot of that and just a tiny bit of this, basically. Your micro versus your macronutrients. So I'm gonna highlight the ones, I've already told you phosphorus is a big one that mycorrhiza will take up. I'm gonna highlight all the nutrients that mycorrhiza are capable of uptaking and transferring directly to the plant. So place your bets. Actually, Dan's gonna highlight them. How about that? They detect stress in the plant and try to alleviate it. So when the plant has a shortage, they try and fulfill that shortage. What an amazing thing. The only reason molybdenum is not lit up too is because I couldn't find any research where anyone checked. But I'll bet they probably take that up too. Dan? So we're going to take a real close look at 16 things on that list, or too much. It would take 3 days to go over all of them. But we're going to take a look at the 4 that I think are most important to farmers, and that is increasing yields, getting free phosphorus, getting the drought tolerance going, and biocontrol of disease and pests. Okay? So Leckberg and Coide in 2005 looked at 290 field and greenhouse experiments. This is called a meta-study. All the experiments aren't the same. They're all very different, but there's special statistical analysis you do for a meta-study that compensates for all the different variables. And so what they found when they did this meta-study is that in general, increasing root colonization caused an increase in yield, on average, 23% across all management practices, all of them. The good, the bad, the ugly. They weren't picky. So that means that AM fungi mean better yields. And if you think back to that list of 16 reasons you should care about them, it's not hard to figure out why they increase yields. Okay. So, free phosphorus. This is an older study. I forget, it's 1979, but I chose it because it's the most typical results. You see this response in these ratios again and again and again in the scientific literature. So we're looking at soybean plants, and we've got their dry weight, no mycorrhizae in black, with mycorrhizae in gray. And this is the dry weight is in grams per plant, and you can see the dry weight is more than 1.5 times. A 30% increase in weight in mycorrhiza versus plants that are denied mycorrhiza is common. So you can see there's not much effect on copper, which is a micronutrient for plants. Copper is toxic to most fungi, so that's not that surprising. Zinc is nearly tripled. That's very common. We are now seeing dietary shortages in zinc because our plants aren't uptaking it like they used to when mycorrhiza were more common. And phosphorus doubled. That is a very typical response to mycorrhizal fungi. They double the amount of phosphorus available. They make unavailable phosphorus available. So they create acids, the plant creates acids, they both create enzymes, they combine those enzymes, and they get more phosphorus that way. Plus, you've got more soil available to you because you've got more surface area. When you add the surface area of the roots to the surface area of the fungi, how much soil is available becomes considerable. And the surface area for uptaking the nutrients increases dramatically, up to 10-fold. Some people say 100 to 1,000. I don't know if it's really that high, but I would not hesitate to say 10-fold. So here's how the relationship works. You've got your corn, or whatever mycorrhizal plant you want. I chose corn because I did most of, my position with USDA was actually funded by the Corn Utilization Council. For 4 years, they funded my research. So the corn is providing sugars to the mycorrhizal fungi, who in turn are uptaking minerals and providing them directly to the plant. And that's the bargain, that's the deal. They're experts, they have their own efficiencies. It makes the acquisition of minerals cheaper for the plant. And it's gone on so long that the mycorrhizal fungi no longer even have the genes for uptaking their own carbon. They've lost them because they depend on the plant for it. Go ahead. Then what do? We dump tons of expensive fertilizer all over our fields, including phosphorus. In nature, phosphorus is very scarce. It's a rare resource, so plants usually don't get very much of it. It's a limiting resource. So this relationship evolved under conditions where phosphorus was scarce, and so all the decisions are made based on the availability of phosphorus. So when a lot of phosphorus is available, the plant says, oh, I don't need to spend anything on mycorrhizal fungi. I'll keep all that carbon to myself, or I'll secrete it through my roots. They secrete quite a bit of sugar through their roots into the soil to keep bacteria and other microbes active around their roots, because that's how they get a lot of their nutrients, from the behavior and life cycle and the life and death of all these things that are in the soil. And they don't send out a signal that says colonize me. So the mycorrhizae stay in their spores, and the plants lose all of those 16 benefits, all of them that they had before, because the decision is a biochemical decision. It's based entirely on phosphorus availability. So if you have too much phosphorus down and you let your phosphorus levels on your soil test get too high, your mycorrhizal fungi are going to slowly die out and disappear. And that's probably a large contributing factor to their loss in our agricultural system. Go ahead. So what happens is we dump our phosphorus. The plant says, I don't need to give you sugar. The AMF can rest in peace. And all that free fertilizer, did you hit that? Is gone. You're buying fertilizer now because you got to do the job of the microbes because you just wiped them out. And they were providing 50% of that plant's phosphorus needs before we involved ourselves in the system and said, oh, I got to make sure I have a big crop, right? So I got to put a lot of fertilizer. More fertilizer does not always equate to more yield. And you never have to forget your profit margin. The more you spend to get that little bit more, the less money you make in the long run, right? Because your inputs become so high. So you got to balance that out. The highest yield is not always the most profitable yield. Now, the seed companies have these contests every year. Who can get the highest yield? Because that sells seeds. Oh, everybody's going to want this guy's seeds. And I learned about this when I first started working for the USDA, and I said, you know what, I'm going to go to one of these. And there was a farm called the Hefty Brothers, and they sell all stuff on ag PhD. Pretty sure they don't have any PhDs. But I went to their farm because you know what they were better at than we were? Getting people to come. We had 300 people at our field day, and they had over 1,000. I'm like, why? What are we doing wrong? Why aren't we bringing people in to hear the science while these guys are bringing people in to sell them stuff? Maybe we should sell them stuff. So I went to their field day, and they brought out this guy who'd had, this huge yield. I forgot what it was, over 400. And they slapped their hat on him and put him in front of a crowd, and he won a big prize, and he talked about it. And there was another guy, everybody was asking, what variety did you use? Everybody wanted what he did, right? They weren't considering that they weren't living in the same part of the country he was. They weren't considering that if they plant those seeds, which have a long maturation period, and they don't have the time for it to reach that, they're not going to get there. If their soil is different, they're not going to get there. And what really struck me about this was the guy lived on a riverbank and was under constant scrutiny by the government for leaching. He was right there on the river. And one of the things that he said was, I couldn't use much fertilizer. Less became more. Less fertilizer resulted in that massive record crop. He could barely put anything in there because it leached directly into the river, and he was monitored closely. And he probably had a lot of mycorrhizal fungi working for him because he wasn't over-fertilizing And he had been putting very little fertilizer for many years on there. And that probably played a big role in his plant's health. Because it's not just about getting the nutrients into the plant. It's also about protecting the plant. You have a little drought period, or even a long drought period. Are you going to lose your crop? Even a small haul is better than crop insurance in most cases. You know, so you've got all these protective mechanisms going on, and you're going to lose them all when you lose your fungi.
On your phosphorus question here, when you've been speaking, when you talk about the numbers, talking about the P1 test, should we be looking at P2 tests as well as total phosphorus? Because isn't some of that free phosphorus coming actually out of the P2 or the unavailable phosphorus?
Okay, so your P1 and your P2 are kind of unrealistic tests to begin with. Phosphorus binds to everything. If you want to test for phosphorus, you need 100 tests to get all the total phosphorus. So we only ever see a fraction of the phosphorus. It's a highly charged molecule. The minute it hits the soil, it's becoming unavailable. It's binding to organic matter. It's binding to other minerals. It's binding to everything. It is just hard to keep it in its free state. It just doesn't stay that way. These tests that are the common p-tests they utilize really harsh acids to separate the P and get it out and measure how much is there. And none of those acids are going to occur in your soil. So that's why I like the Haney test. It uses the same acids that plants and microbes secrete. And their recommendations usually are lower than those based on the P1 and P2. And I think it's a good test. I like it. A lot of people who have tried it like it. Some people have said they don't like it. I've had people tell me there's not enough research behind it for them to trust it. But in general, from all the people I've talked to who have used it, most of them, the vast majority, have said, I had really good results relying on this test. Yes?
Wendy, when you talk about that range of 1 to 200 parts per million on the P, is that coming directly from the Haney test?
No. I actually didn't ask. I'm going to phone Ray and ask him what tests they used, where he got those numbers from.
And the reason I ask that is I'm trying to correlate something that we would be familiar with versus, you know.
Right. So I'm guessing that it probably didn't. I got to find those papers and see which tests they ran. It's probably the standard, you know, P1, P2, your Malick tests. But the point here is not to overdo it. Start inching things down. If you have really high phosphorus and you suddenly stop putting in what you were putting in before, your system isn't ready to absorb that change. Your microbes aren't there to compensate. So you have to make your reductions gradual. So as things reduce, other things that can tolerate those levels will come in. You reduce a little more, and other things, you know, because different things have different tolerance levels, will start to move in. So you got to start gradually reducing. And you'll see that a little bit less doesn't seem to affect anything. So no more free phosphorus, because P is like a magnet. It binds to everything. Less than 20% of the phosphorus that you put in your field actually gets inside your plants. Less than 20%. That's huge. And I once asked a fertilizer salesman, I said, well, if less than 20% is getting into the plant, why do we keep putting more and more in our field? And he goes, I go, where is the other 80% going? And he said, oh, it's there. It'll be released next year. So next year, I don't need to add more phosphorus, right? In fact, for the next 5 years, I don't need to add more phosphorus. No, you gotta add more. Why? Oh, you just gotta. That was his explanation. You just gotta. So clearly we need a better understanding of what phosphorus is doing in our soil and what the benefits are and how much is going to benefit our crop and how much is hurting the biology in the soil, which hurts the crop in the long run. So less than 20% is going to get actually into your plants and into the tissue of the plants. The rest of it runs off. It gets bound if you have erosion. It's blowing away. Do you know we have dirty 30s, dust storms going on every year now in the United States? Well, not just the United States, around the world. Because we've lost the soil quality. We've denuded the soil. If you can see your soil, it's eroding. You never want to be able to see your soil. It should always be covered. And it winds up in the water. Ultimately, that's where it goes, and it kills fish. I personally love to fish. I don't like those kind of fish, though. And honestly, financially, it's like putting 1 gallon of gas in your car. Would you flip that? And using the other 4 gallons to kill fish. That's what we're doing, literally. And there's another name for that. Well, that's what we call it when we kill the fish, eutrophication. I'm not going to explain the process. It's kind of lengthy, but I call it throwing away your money. You are just burning that money. It's not what you want to do. You want what you put down, you want to put down less, and you want to keep it and have it work for you. You want to keep it in your soil. So if it's running away and blowing away onto your neighbor's soil, it's not benefiting you. You're wasting your money. So AM fungi mean money saved on chemical fertilizers. Now we're bringing our cost to do business down. Go ahead. Drought tolerance. All right. I really struggled as an undergrad and an early graduate student to understand this because these guys are so small you need a microscope to see them. And I pictured their hyphae like straws, you know, little straw, big straw. How much water can they really uptake? They're microscopic, come on. But they don't bring it up like a straw. They absorb it through the surface area, and they increase the surface area that is servicing the plant tremendously. Go ahead, Dan. So Riz Lozano and Ascon in 1995 made this really clever experiment to try and figure out how much water the mycorrhiza fungi could really take up. So they took a pot and they put compartments in it and filled them with sand. So they used a 50 micron nylon mesh. Think of it like a nylon stocking, only smaller, finer mesh than that. And it's so small that the roots can't get through it. But fungal hyphae are much smaller than roots and even smaller than root hairs. And they could pass through that nylon mesh. And then he had, this is the hyphae only. This is the root compartment, but it's root and hyphae. And then the hyphae grow down, and you have no roots in this compartment. And then they grow down to a third compartment, and they injected the water at the bottom instead of watering from the top. So all the water that went to the plant had to be transported there by the hyphae. And he inoculated one with Glomus fasciculatum, another with Glomus deserticola, and to make sure that any growth differences weren't attributable to less P, because even in sand, mycorrhizal fungi are so good at scavenging phosphorus that it might make a difference in the plant growth, he gave the control fertilizer that contained phosphorus, just to compensate for the nutrient uptake abilities of the AMF. So, place your bets. Yeah, there we go. 150% more fresh weight than the fertilized control. 215% more fresh weight than the fertilized control. Deserticola, desert. This mycorrhiza came from a desert. Now let me explain something to you for people who are exploring products. If the mycorrhizae aren't native to your soil, they're not going to compete with what's already in your soil. And in a pot is a prison. A pot is a prison for a plant. And if you're in a prison and all I feed you is Brussels sprouts and you hate Brussels sprouts, but your choice is Brussels sprouts or starvation, what are you going to do? You're going to eat Brussels sprouts. So in a pot, you can get any plant to associate with any species of mycorrhizal fungi, for the most part. But in the field, that's not what happens. In the field, we find that certain plants associate with certain species. So almost every mycorrhizal product out there contains Glomus deserticola, and it is like throwing a polar bear into the desert. It is not going to colonize anything in your field. Furthermore, they add fertilizers to make you think something good happened. So be aware, trace minerals and other things. to trick you into thinking that the mycorrhiza did this job for you. You need to be aware of that. Be savvy. So AIM fungi save money on losses due to drought. I suspect these varieties of corn that they're breeding now, the ones that are drought tolerant, they're probably increasing the symbiosis or maintaining the symbiosis. And we are seeing, in all my years of study, I never saw corn that was non-mycorrhiza. We are now seeing non-mycorrhizal corn being released. And I think what's happening is because of the merging of the chemical companies and the seed companies into one, the seeds are being grown under high input systems, right? Because if you're a chemical company, you want to sell a lot of chemicals. So they set everything up and they spray everything and they use all these chemicals. And what happens when you do that is the mycorrhizal fungi, under these high input systems, not only do they die out, but there's one group called Rhizophagus. Although usually on the bottles it's labeled as a glomus species, Rhizophagus intraradices. It lives inside the cell, it makes its spores inside the root. That's why it's called intraradices. It actually bursts out of the root, destroys it when the plant senesces. So it's not killing your plants. But it is a nitrogen pig. It loves nitrogen. When everything else is dead, that guy is what's left. And out of all the different groups and species of mycorrhizal fungi, it's the only one that can be, that to my knowledge, can force colonize under high input systems. And now they're selecting, because it becomes a yield drag under those conditions, under those high input systems, they are now selecting against the symbiosis, and we're losing it in our crops. They're breeding it out. And I think it's important that we start finding out which varieties it has been bred out of and making that information available. There is one company that I know of that is actually doing their plant breeding under low input conditions and no-till, which are the conditions that are best and most economical and most profitable for farmers. I'll have to find them. I think it was, make sure I don't get them mixed up with the soybean company, Emerge. There's Emerge and, now Spectrum corn. And they are breeding under the conditions that a lot of you are growing under. So it wouldn't hurt to give them a call and see what they're doing and what they have, and if they have varieties that are good for your area. I'm not familiar with their whole range of products. But I talked to one of their founders one day on the phone, quite by accident, called him up looking to buy corn seeds, and started discussing. It turned out he went to the same university I had, and we just started talking about stuff, and he told me about how they're bred. And I'm like, wow, nobody else is doing that. That is great. Okay, next slide. So Biocontrol Agents. I went through all the research, and I picked out a bunch of stuff that was common. I did this when I was at the USDA, so most of these diseases are common in North Dakota, as I borrowed from the university there. But as I've given talks across the country, most people have seen these diseases. So they're common in a lot of places. And these diseases have been found to be suppressed by mycorrhizal colonization. I had a friend tell me they had planted cover crops, and they had chemically terminated the cover crops and planted corn, but they were a little late on planting, on terminating the cover crops in one side than they were on the other. And she said the difference between the two fields, which had the same variety, planted at the same time, same soil type, the only difference between the two fields was that she had terminated the cover crops a little earlier on one side, and then waited a week or two before they terminated on the other side. They planted everything at the same time. She said the difference between those crops was like day and night. What happened? She said the only thing I could think of was maybe it had to do with the mycorrhizae. So she sent me some plant roots. Said, could you tell me? And she didn't tell me which was which. So I cleared and stained the plant roots, and I looked for colonization. And what I found in one set of plant roots were mycorrhizal colonization. And in the other set of plant roots, I found colonization by a pathogenic fungi. You almost never find them occupying the same roots. In all my years of studying mycorrhizal fungi, I have never seen mycorrhizal fungi and pathogens sharing roots except once. Once. And I've looked at thousands of slides, I guarantee you, thousands. So it's really rare because they don't like each other. And that one time I saw it, if I had probably checked it a week later, I'll bet the other guy would have been kicked out by then. Chemical warfare. She didn't tell me which was which. I sent her the results. She said, wow, I didn't think we'd get any colonization. And the one that had the pathogen didn't have any mycorrhizal colonization. She said her phosphorus levels were pretty high, so I wasn't expecting colonization on either. So colonization kept that pathogen out, and because they terminated the cover crop late, the mycorrhizal fungi had enough time to proliferate. And if there's just nothing green there, they die. So companion cropping, folks, is the way to go. If you can find a low-growing clover to plant between your corn, you have gained. You're not going to get just the nitrogen, but they're going to support mycorrhizae. Clovers are great mycorrhizal hosts. Corn is a great mycorrhizal host. They host many, many different species of mycorrhiza. So when your plant is colonized, it's not just one species. It's got dozens of species colonizing it. And look at this list. Root rot, fusarium, take-all disease, root rot disease complex. These guys are nematodes. So once they damage the root, all sorts of other things have an opportunity now, right, to get in there that they couldn't penetrate the plant's defenses without help. Phytophthora, Pythium, Rhizoctonia, Verticillium, common plant diseases. Mycorrhizal fungi protect plants from all of those to a great deal. I did an experiment with sorghum because someone asked me, can you solve the, they had anthracnose on the sorghum. What a problem, huge problem. So I set up an experiment in pots and I colonized one and didn't colonize the other. The plants without the mycorrhiza got about this tall. The fusarium just ate them up. I mean, just completely wiped them out. And then the other plants were in less than 4 weeks, 8 times larger. They still had fusarium, but it was like little pinpricks. It could get started, but it couldn't spread. So the colonies didn't spread and become a big mass and just destroy the plant. So it's not going to necessarily eliminate it 100%, but it's going to eliminate it to the point that it doesn't matter, that it's not really affecting your yields. Also, I've been told that if you are concerned, for people with cattle particularly, with some of these fungal diseases, creating toxins that can kill your cows, that using roasted grains will kill a lot of these toxins and denature them so they can't function. So it's a consideration. I don't have any research on it. It was a guy who'd run cattle his whole life. This is to show you kind of how the mycorrhiza defend themselves. Could you flip that? And here comes a nematode. And the nematode wants to chomp on these roots. So all of these plants are interconnected. So each color represents a different species. Mycorrhizal fungi can fuse their hyphae together, but they have to be the same species. They don't cross species. So all the yellow guys are one species, all the red guys are one species, all the blue guys are one species, and they interconnect their hyphae across huge networks. And when this guy comes and chomps on this plant sends out stress signals. And the mycorrhizal fungi, before the plant, also send out signals. And the mycorrhizal fungi can take over the plant's signaling response and amplify it and turn it on and off. Say, whoa, this is a big problem. We can't let this guy eat our host. Start sending out these defensive chemicals. So the plant starts making chemicals to kill the nematodes. But it's too late for this guy. He's going to get chomped on a little bit, but that signal goes out through these hyphal networks and reaches all the surrounding plants before the nematodes reach them, and they activate their defense mechanisms. Now they're no longer tasty. They produce jasmonic acid and other chemicals that the nematodes don't like. So the nematode chews on this for a little bit, and then he wants to go on and chew on something else. And he just has to go further and further and further to find a naive plant that doesn't know he's coming. So this reduces nematode damage. Okay. How much? So it's AMF versus nematodes. And I just was amazed by this because a nematode is an animal. So I was thinking at the time when I was a student, How can a fungus really defend itself against an animal? Well, you poison it. Go ahead. So AMF versus the soybean cyst nematode, the damage was reduced 36%. Next one. In a review of AMF nematode interactions that spanned from 1996 to 2004, Hole and Cook, they did a meta-analysis, and they found that AMF are most effective against the root knot and cyst nematodes, and the average damage reduction was 22%. They're particularly effective against stunt nematodes, where they cause a 21% decrease in nematode numbers. The amazing part about that was they were almost all female nematodes that were being targeted. So they had found a weakness that they could exploit and produce some sort of a toxin that killed female nematodes. Now, I'm a biologist, well, an ecologist, but that's a type of biology, and I know that when biologists want to control a population, they go out and shoot the females, not the males. So that was just amazing. So, migrating nematodes. The mycorrhizal plants were more than twice the size of the non-mycorrhizal controls. So that's some of the mechanisms by which the mycorrhizal fungi protect and support their hosts. Now we're going to take a look at some of the management decisions that affect the things that are in your soil, particularly the mycorrhizal fungi. And see these tractors? See all this soil in the air? Raise your hand if you have never seen that. Oh, not one hand. Okay. I'm not even a farmer and I've seen that. You know? This is not where your soil belongs. Okay? It's doing you no good in the air. The topsoil that gets the most disturbance when you till is where most of the nutrients are. The nutrients aren't down deep, they're near the top. That's where they begin. So in your soil profile, if you took nutrient measurements and you went deeper and deeper and deeper, everything declines as you go down. So where do those nutrients come from and why are they up there? Because the deep-rooting plants bring them up, and when they die and rot, they return the nutrients to the surface. So the nutrients are always being cycled from deep to shallow, deep to shallow. They're moving. So what happens when we till? Let's take a look. This is a corn seed. You know, it wasn't until I worked for the USDA that I'd ever seen pink corn because of all the, I want to say crap, all the crap they coat them with. I mean, the poisons, herbicides, fungicides, and what really got me was the seed protectors. The seed, you got to protect the seed from all the other stuff and from the herbicides and stuff you're gonna put on there. I took some seeds that were sent to me from a seed company for an experiment. I'm not allowed to name names, but they gave me the seeds with 1 coating, 2 coatings, and fully loaded, and only the protectant. And one of the things I noticed that seeds with protectants germinated later. So those seed protectants suppress germination. The plant has to get past that, or maybe wait for enough rain to wash it off or dilute it enough for them, or for the microbes, if there's any left in your soil, they can get there, because that is a really, really toxic zone. The good news is mycorrhizal fungi are very resistant to seed coatings, because we tested mycorrhizal fungi, and I had a student do a really large-scale test, and mycorrhizal fungi are not really affected much by those seed coatings. There you go. So your plant germinates, and these little colored lines are the mycorrhizal fungi colonizing the plant as it grows. We never plant just one seed. We plant a bunch of them. And the roots interact, and the fungi interact. And I just drew this randomly and did a copy-paste, so there's nothing special going on there. And then, go ahead, we harvest, and this network remains behind in the soil. And that network can be reactivated. It goes dormant in the winter, but we can turn it back on. No one's sure whether it dies and the new hyphae race through there and absorb it for minerals, but you got to figure if the path is already there, it's going to take less energy to run down a path than to hack a new path through the jungle, right? So it's saving energy when the new mycorrhizal fungi germinate and come next year. They're going to reuse those paths. Ed?
Wendy, are you suggesting that the hyphae can survive without a host plant for a period of time then?
They can go dormant, yes. Many mycorrhizal fungi can reproduce from hyphal fragments if they're large enough, depends on how big they are and what species it is. So most of the glomus can colonize from a hyphal fragment. Whereas the really big group called Gygospora, they make the largest spores, they cannot do this from their hyphal fragments. So it's a trade-off.
Related have a response from temperature?
So the question is, does a dormancy have a temperature response? I'm not sure I understand what you're asking.
Well, I mean, will they survive longer in dormancy with colder weathers compared to warm temperatures, warm soil temperatures?
Well, so the life cycle, so he wants to know, will they preserve themselves in the soil longer when it's cold? And actually, if the soil is undisturbed, they can remain intact for a long time. And the spores can live a long time too. So if the plant gets to run its full life course to maturity and senesce, when it starts filling its seeds, it's reducing the nutrients that it's been sharing with the mycorrhiza. And the mycorrhiza says, oh, that's my signal to make my spores. So their life cycles are interrelated and tied together. So you get a combination of spores and hyphae in the soil. I store my culture collection under refrigeration. When I first started working with these, I was told 5 years is the maximum, and then they're going to die. But I have personally revived cultures that were 8 years old, and a couple that were 10. And then one day I was talking to another mycologist who also studies mycorrhizal fungi, and she said, you know what? These researchers brought me a piece of fabric from a tomb, a mummy's tomb, and it had some little tiny spores on it, and they were wondering if they were mycorrhiza. She said, I didn't think anything would happen, but they did kind of look like mycorrhiza, so I laid them gently on some plant roots and planted it, and they grew. So I had been bragging about 8 years. She had 8,000 years. So I lost all bragging rights right there. 8,000 years. So if the conditions are right, the spores can last a very long time. But also one of the things I've noticed just from maintaining a culture collection of these is if I chop up the soil in the pot that I grew them in, they don't last as long as if I just take the whole thing, soil and pot intact, dry it down, and refrigerate it. Those guys last a long time. So disturbing the hyphae affects the lifespan of the spores too, which surprised me. Okay, so now what I did here was I erased all the hyphae that weren't directly connected and in contact with the root, and this is what was left. Go ahead. And now here's another plant that has no mycorrhizae associated with it. The next slide. And that's the difference in the volume of soil available for nutrient and water uptake. This is the big edge that mycorrhiza give to plants. More surface area for uptaking everything they need. Okay? Now these lines represent breakage points of the hyphae. So now I basically cut the hyphae wherever I drew a line and then erased anything that wasn't still in contact with the plant. Okay, next one. And that's what we got. Now we're being colonized by fragments instead of tapping into a network. Plant still gets colonized and you still get a lot of benefits, but you've lost the benefits associated with the network. Can I have the next one? So the thing that mycorrhizal fungi are most famous for is the uptake of phosphorus. And in corn, your yield potential is determined by how much phosphorus you can actually get into your leaf tissue. Adding phosphorus to your field after the leaf stage, after the 5-leaf stage, will not increase your phosphorus to the plant. It's already made its decision as to how many rows of corn and how big the kernels are going to be and how long the ears are going to be. All those decisions are biochemically made based on phosphorus. How much phosphorus did I get early? How much can I store and then later shunt up to those ears and make them grow and fill them out? So that decision happens early on in the life cycle of the plant, and you just shot yourself in the foot on your yield potential. because that network is no longer there, and you lost that extra volume of soil and the early P, the early phosphorus to your plant. So you're still going to get the other protections, but your yield is going to suffer for having destroyed that full network. Right. Furthermore, when the mycorrhizae don't find that network, they start regrowing the network, right? They got to get out there into the soil. So instead of having that low energy network that they can race through and reestablish and repair and fill and resorb and get to work really fast, that's all gone. So instead, they have to rebuild that network and regrow it. That means they need more carbon from the plant to do that. So you just increased the carbon cost of the symbiosis by destroying that network. You have a question?
Does that happen in time before they reach the V5 stage?
Does what happen in time?
That recolonization.
Oh, the recolonization. Oh, yes, yes. So how old is a plant at the V5 stage? How many weeks is that? It's 2 months, a month? They're usually fully colonized in 30 days from germination.
Does the phosphorus level change that? You know,
In the soil?
does high phosphate slow that process?
You will not get colonization if the phosphorus levels in your soil are too high. The plant will say, I don't need mycorrhiza, and reject colonization. So you won't get any colonization, you won't get any of those benefits under most conditions. You might have some rhizophagus that can survive in there, which can, under high input systems, be a yield drag. Usually when I look at a lot of different farm soils, and I've looked at a lot, believe me, and I look at spores and see who's there and who's absent, if I see the gigaspora, I call the farmer and ask him what he's doing because I'm excited. He's preserving those guys that don't propagate from hyphae, so he's getting everything out there. But the last thing to go is the rhizophagus, which can be a yield drag. And if you're getting a lot of nitrogen in your soil, you're going to have a lot of that guy in there. Because every time you change your soil conditions, you alter who's colonizing. And the two main players in this, the guys that make the most intensive, massive networks, are those really big spores, the gigaspora. And the guys that are the last to go, and if I find nothing else, the last thing to go is always the rhizophagus because of the nitrogen we put down. So you want to be kind of at the other end. You want that diversity. And tomorrow, or later today after lunch, I'm going to talk about diversity, and you'll get a bigger grasp on how it's important. But remember that it's the diversity of mycorrhizal fungi that give you the diversity of those benefits. Some species are really good at one thing. Like I do all of that to some degree that's on that list, but I'm really good at killing that one disease. And this guy over here is really good at killing another disease. And this third guy is really good at watching out for nematodes and sending out those signals. So you want that diversity. You want that diversity because it's working for you.
Does the form of nitrogen affect what nematodes are present? If you're using, let's say, UAN versus NH3?
Oh, that's a great question. Does the form of nitrogen affect which nematodes or mycorrhiza? You said nematodes. I wasn't sure. I don't know. Now I have to do work. I got to find out the answer to that. I don't know. I've been studying nutrients in hydroponic systems, and I know in hydroponic systems, well, too much nitrogen will burn out everything, but also most of the nitrogen should be nitrate, but you want a little bit of the NH4 of the ammonia because it actually stimulates uptake of the nitrate to have a little bit. So you actually want both. And the ratio that the book I'm reading right now recommends is between 5 and 25% ammonia and the rest nitrate. That's the ratio they recommend between the two forms of nitrogen. So how does soil respond to tillage? Well, your AMF and other microbes decline. When your AMF decline, all your other microbes tend to decline. All your other beneficials, fungi and bacteria, decline with them. They no longer have those hyphae. They no longer have the protection. They're no longer getting as much sugar because the plant actually shunts, think about this, 30%, I should say up to 30% of all their carbohydrates directly to their AMF. That is like me saying, I would like all of you to give me 1/3 of your paycheck every month. I'm going to do really good things with it. I'm going to help you with it. I'm going to spread the word. May I? May I please? Yeah, but I'm not getting 1/3 of everybody's check for it. But think about that. That's a huge, huge exchange that the plant has control over. The plant can actually dissolve arbuscules that aren't producing. So this arbuscule isn't bringing up enough nutrients. Those hyphae have used up whatever nutrients there were, so I don't need this one anymore, and they dissolve them. The arbuscules last about a week. Then they've taken up everything they can reach, and new hyphae are being formed and finding new spots. They're searching the soil for the nutrients, right? So the plant is controlling this relationship and how much sugar it gives. And to the plant, the relationship is so important that it will give up to 1/3 of its carbon to feed directly to the AMF without the competition, not out in the soil, not secreting into the soil, through the arbuscule, without competition from other microbes, directly to the AMF. That's how important the relationship is to the plant. So our goal is to reduce the cost of the symbiosis. Plants that are colonized also increase their photosynthesis to compensate. So the 30% doesn't mean that your plants are getting smaller. They're actually increasing their photosynthetic rate to compensate and to create the extra sugars. They secrete some through their roots. They feed a lot directly to the AMF. And the AMF secretes some of that sugar too through their hyphae. Because they're all, the hyphae, the plants, the fungi, nurturing the bacteria in the soil and the other things that come to the roots to live. Because the plant knows that's how I stay alive. These guys are cycling nutrients right here, and they need to cycle it right here where my roots are. So, when you have tillage, you lose your microbes. It creates erosion. It creates compaction. You're going to have a hard pan right underneath where that implement passed. You introduce oxygen into deeper layers of the soil. This causes aerobic bacteria deep in the soil to suddenly have a population boom because there's oxygen down there. Where before, they were hardly any down there. They were just a tiny, tiny bit of the population, barely making it. Now, they're getting oxygen. What are they going to eat? Your carbon. So you get a carbon loss because you introduce that oxygen into your deeper layers. They chew up that carbon and convert it to carbon dioxide. It's leaving your soil. Reduces your water holding capacity. You're breaking up, soil has structure, it has layers. Things are organized, fine particles down here, coarser particles even deeper, organic matter near the top. There's layers, they call them horizons. And soil has structure, and plants have evolved to live in a structured environment. But now when you churn all that, not only are you altering everything, losing your carbon loss causes a loss of water holding capacity, but compaction is also causing a loss of water holding capacity because you're breaking everything up, and then a rain comes and it all compacts down. Predatory insects are killed. Not the pests. They're not killed by tillage. Just the predators. How does that happen? That's kind of weird. Because Mother Nature says, all you predators, you can't emerge from the soil where you live until there's food for you. So the pests come out first, and the predators come out later. And the timing of our tillage usually falls between those windows. So the pests survive and emerge, and we kill everything that would have hunted them down. And it plants weeds. Do we take our seeds out and just throw them out there onto the field? Or do we carefully make a little trench and drop the seed directly into it and have a little tool that pats the soil down? Why do we cover the seed? Germination. You need that soil contact. You need that moisture coming up against the seed. Also, have you ever seen birds follow your tractor and pick up seeds? Seeds that fell. Sometimes they'll even try and pick them right out of the soil, right? So there are lots of seed predators, not just birds. There are insects, many, many insects are also seed predators. When a seed is laying on the ground, it's easy to find for seed predators. So when you bury it, you hide it and protect it and nurture it. When you till, all those weed seeds that have blown across your field that you have not seen and are unaware of, but they are landing there every day. They're raining down onto your field, waiting for a chance to germinate. Then you till and you plant them. You propagate your weed problem. Now, at the USDA, we had an amazing weed scientist. His name was Randy. Randy Anderson. So if you ever get a chance to look for any of his papers, look up Randy Anderson. Really shy guy, very quiet, very withdrawn. My boss was, you got Randy to come out and go to a talk? How did you do that? I used to go pester him. I didn't let him be shy. I wanted his knowledge because that guy was smart. And he developed a system of cover crop management that defeated weeds. He said, in 5 years, you won't have a weed problem. It's a process because you've got a seed bank of weeds that has built up over the years buried in your soil. And that seed bank has to be depleted. But if you alternate your cover crops, warm, warm, cool, cool, warm, warm, cool, cool. 2 seasons of warm season cover crops, 2 seasons of cool season cover crops. What happens is that your cool season cover crops defeat the cool season competitors, and your warm season cover crops defeat your warm season competitors. They compete against them, cause a decline in their numbers. Because you're alternating, you're wiping them all out, and you're using up that seed bank. So you're out there spraying what you see, and year by year by year, if you stop tilling and alternate your cover crops, you will defeat and smother out and outcompete your weeds. And then as long as you don't go back to tilling, you'll probably never have a serious weed problem again. Things will still blow in, but as long as you're keeping that up, they're never going to get a good grip and become enough to be a problem. So I love Randy. That was such an eye-opener for me. I was like, wow.
So are we kind of suppressing the effect of that if we use the shotgun mixtures, cover crops?
So are we suppressing weeds with a shotgun mix of cover crops? Are we altering that effect?
Yes, that
Yes.
you're reducing the effectiveness of weeds.
You're probably having a different effect. Because when you use a shotgun effect, what's going to happen is conditions are going to determine what comes up and what remains dormant. So there's always a benefit to cover crops because in weed control because they're competing regardless. But it's not the same as going warm, warm, cool, cool. I don't think. I'm not especially a weed scientist, and I don't think anyone's ever tested and compared the two. But shading out your weeds and out competing them that way is a good way to keep them down. Alfalfa, if you've got cattle, 5 years of alfalfa wipes out weeds, man. They can't get through that. And alfalfa is a great mycorrhizal host. Okay, so if you have compaction problems, there's 2 ways to recognize them. The water is on the soil and not going into the soil. That's a compaction problem, usually. Unless you have a very, very shallow water table. So you're not getting good penetration of your water. You should take a pipe out there, a clear pipe, and mark it off in increments and pour a gallon of water, mark your 1 gallon into it, plant it down good, and time how long it takes each inch of water to go down. And then watch every year as you start using cover crops how much more water infiltrates into your soil. I had someone ask me about tiling once, and he said, what do you think about tiling? And then I said, oh, I think tiles should be broken and destroyed. We shouldn't be tiling everything. And he's like, oh, no, I'm loud, right? So he thought I was yelling at him. I said, oh, I had to apologize. I said, here's the thing. Tiling is a Band-Aid on a symptom. The symptom is poor water infiltration. But the problem is not because you don't have tiles.
Okay, we got a hold it right there. No, I got that comment.
Oh, comment. Yes, comment.
A lot of our soils without tile, we would have water up to the surface maybe throughout the entire summer.
Yeah, a lot of areas were drained for farming. That's a different situation than compaction. That's a different situation, yes.
Okay.
I acknowledge that.
tiling is needed in this country.
But you gotta be careful. You've got to analyze what's going on. If you're in a lowland and have a high water table, you're gonna have water sitting up there no matter what.
Yes.
Yes. Now that is different than having a compaction layer. So deep rooting plants will help with this. They'll create tunnels through compaction zones if that's your problem. And I love the dialogue and the arguments, not arguments, angry arguments, but the discussion of all of this. No, we need that. That's what we should have. So let's take a break and let people walk around and talk to each other and discuss this problem.